Okay, good morning. Let's have a class. Before we start to talk about the lab, I would love to spend it about like 20 minutes to go over the complete aerobic respiration and also the ATP calculation. And our videotape put on the YouTube channel, you can review anytime. You can take a big picture of the blackboard for your outline to study. Okay, so this is we call it aerobic respiration, the ATPs calculation. Now, of course, we're gonna go all these basic chemicals, okay? So we started to talk about is glycolysis. The first chemical everybody should know very well is glucose. So you should know glucose very well. 6 carbon sugar. Okay, HOH goes here, HOH, then goes here, OH, H, then OH, H, and top H, CH, 2OH. This is glucose. And you should know how to label the carbon. That's important. Carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6. Therefore, it comes out with a next chemical. What is that? Is glucose 6-phosphate. Okay, so let's draw it. Right here, I'll draw a little bit down here. This is the same. You can copy it. Exactly the same. So you know the first one, basically you know the second one. So we just copy it. HOH, OH, H, OH, H, H, then here CH2O. Then I'll change the color. That's a phosphate. Okay, that's why we call it glucose 6-phosphate. You can just write a P there. Now, the first thing, where that phosphate comes from? It comes from ATP, originally stored in the bacterial cell. So ATP hydrolyzed become ADP and release the phosphate. And that phosphate is used. So here, it is actually used one molecule of ATP. Okay, glucose 6-phosphates. We're gonna go down next one. It will be changed the morphology, but the chemical compounds are the same. So we call it isomerization. So what is that? The next one is fructose 6-phosphate. So what it looks like? It's a little bit changed the morphology. Looks like this. CH2, OH, OH, AOH, H goes here, H, OH, H, CH2, O. This is a phosphate. Okay, be very careful. The tricky thing is here. This 5 carbon is a H, is not hydroxy group. And I actually made a mistake when I draw on the blackboard on the lecture. But in the exam, it is okay if you miss occasionally one chemical. Okay, that's fine. And don't worry about these OH up and down. This is just a different position, so it's fine. But be careful. This is a H, okay? So fructose 6-phosphate. Okay, then we go down. Become what? Fructose 1,6-biphosphate. So let's draw it. That's the same thing. CH2O. It's already called a 1,6 biphosphate. So one position you have one. So we do OH here, OH, H, H, OH. Then go down here is H, CH2O. This is a phosphate right here. Okay. Now, since you have another phosphate is there, where this comes from? Again, ATP hydrolyzed become ADP and phosphate. Okay, so this phosphate added up here. This name we call fructose 1,6 biphosphate. When you see bi, which means there are two phosphates. Okay, so here what we talk about is glycolysis, this line. So from glucose goes to fructose 1,6 biphosphate. We call this is the investment stage because instead of generate ATP, we used 2 ATPs. Okay, now we're gonna go to this branch later on. So let's draw on the top. So fructose 1,6 biphosphates. Go here. We are gonna be a 6 carbon sugar become a 3 carbon sugar. So you know the enzyme here is called aldolase. 6 carbon become 3 carbon. So the first one, CHO, CHOH, CH2O phosphate. Okay? Every time the phosphate here, I'll just use a red one. So what is this? You see CHO, that is aldehyde. Glyceraldehyde 3 phosphate. So this is glyceraldehyde 3 phosphate. Okay, now in your biochemistry class, there is also a transition chemical. We don't talk about here too much. But if you know, you can draw it. If you don't know, that's okay. Which is become CH2OH, CH2OP. That is called dihydroxyacetone phosphate. It's a transition step. Nothing happens. Okay, now we're gonna go here. This step here, I said, is a little bit tricky. What happened? They will become COO. Then you have a phosphate. Then you go here, CHOH. Then you go CH2O phosphate. This is what we call is 1,3 biphosphoglycerate. Now, where is that extra phosphates comes from? This comes from a free available phosphate that goes here. And do not forget, there is an important thing happens is NAD reduced to NADH2. Now, in your biochemistry class, which is said NADH plus H, in my class, it's okay. You just write the NADH2. Okay, Dr. Lam will tell you it's different, but mine is okay. You just write it easy. NAD reduced to NADH2. This is the key stuff here, so we mark it. Okay. 1,3 biphosphoglycerate. Go down. We'll have another chemical, important. What is that? COOH, CHOH, CH2O. That is a phosphate right here. This is 3-phosphoglycerate. Okay. Then this is what's gonna happen. Since above is 1,3-biphosphogycerate, next one is 3-phosphoglycerate. Obviously, there's a 1 phosphate is gone. Where they go? It is donated to ADP, so generate 1 molecule of ATP. This is a key thing here. And we also call this generate is substrate level phosphorylation, because the phosphor transfer power of 1,3-biphosphoglycerate is around, we know that is around like 61, and ATP generate phosphate become ADP combined with phosphate become ATP, the power is about 49. So, the power is about 31, so it's more than that. That's why they can go through substrate level phosphorylation. So that is 3-phosphoglycerate. Then you go down this one. This is relatively easy, not much stuff. You move the position, so what we become is 2-phosphoglycerate. Okay, so we go down here, is CH2OH, that is 2-phosphoglycerate. Okay, then what is going to be the next step? Goes here. We say it's going to lose 1 molecule of the water, so we call it analyzation and need an enzyme called analysis. So what is going to become is COOH, CO phosphate, and then double bonds CH2. This is phosphoenolpyruvate. Okay, phosphoenolpyruvate is a key thing, because the next one is a major step right here. Will generate a very important chemical, is COOH, C double bonds, CH3. What is this? Pyruvate. Since you have this phosphate, the phosphate is gone in pyruvate, so where this comes from? Where this go? Another ADP with phosphate generate ATP. That is also a substrate level phosphorylation. Okay, this is an important guy. Now, some of you said about the enzyme, we don't require you to put the enzyme in the exam, but if you want to know, we could go over something. Glucose become glucose 6-phosphate. This is in fungi, is a hexokinase. We mentioned about that. That's what you learned in your biochemistry class. In bacteria, I said it's not using hexokinase. It's going through group translocation. It's a little bit from biochemistry. This is isomerization, so this is isomerase. Simple. And you go here, you have a fructose 6-phosphate become fructose 1,6-biphosphate. That's fructose phosphate kinase. Okay? And here we already mentioned aldolase. Then here, glyceraldehyde phosphordehydrogenase. Hydrogenase, because it's removed the what we call hydrogen proton becomes NADH2. Okay, when you go here, glyceraldehyde, 1,3-biphosphoglycerate becomes 3-phosphoglycerate. That is another kinase. phosphoglycerate kinase. 3-phosphoglycerate become 2-phosphoglycerate. This is we call it a mutase. Now, obviously, 2-phosphoglycerate become phosphoenolpyruvate enolase. Yeah, is that right? That's not difficult. The last one generate pyruvate kinase. Okay, so that's not difficult. So glycolysis is finished. This part. Then we're gonna go here. The cycle is TCA cycle. Okay, so here the whole thing is glycolysis. So we go TCA cycle right here. So pyruvate we're gonna write on the top. Let's just draw right here. Pyruvate. Okay, so pyruvate, COOH, CH3. First of all, it's going through a transition step. Losing carbon dioxide also happened is NAD reduced to NADH2 and become a very long chemical actually, but we don't talk about that too much, is acetyl-CoA. So this chemical is acetyl-CoA. Okay, since this cycle, we call it citric acid. We call it a citric acid cycle. Therefore, you should know absolutely what is structure of citric acid. So we go here. Let's draw these one by one. What is citric acid? COOH, CH2, COH, COOH, CH2, COOH. That is citric acid. And I want to say here is, in my class, because we talk about bacteria, a little bit different from fungi and animal and human being. You talk about the citric acid cycle, the next one is cis-ascornic acids. We don't talk about that. We directly go to isocitric acid. So we skip one chemical. Okay. What is that? This is going to move down. Hydroxy bond is going to move down. So COOH goes here, CH, goes here, CH, COOH. So you go here, COH, H, and COOH. Isocitric acid, there's nothing happened, just change the position. Then you go here. This is a big step. What happened? Lose one carbon dioxide, happened NAD reduced to NADH2. What is that one? What's the name of that chemical? COOH, CH2, CH2, C. This is a ketone, something like a ketone, then COO plus. I will write the H there. The name of this guy, alpha ketoglutarate. Alpha keto, see, look at the keto, glutarate. Okay, this is the key. NAD reduced to NADH2. Okay, now alpha ketoglutarate, a little bit flat one here. You see the structure here? Continue goes to lose carbon dioxide. Continue happen is NAD reduced to NADH2. So becomes COOH, CH2, CH2, C double bonds, and SCOA. This SCOA come from cell membrane system somewhere. So that one called succinyl-CoA. That's a chemical. Okay. Then you go flat again. Then become COOH, CH2, CH2, COOH. That is succinate. And don't forget, here there is another important thing happen. is instead of we have ADP become ATP, we have GDP become GTP. But I said this is the equivalent of ATP. So GTP at this moment is same as ATP. Okay, succinate generate. When you see succinate generate, you see these chemicals right in the middle. That's not good. It's gonna lose somehow. So how they go? They go the circle like this. Lose H proton will donate to FAD, become FADH2. That's following adenine dinucleotides. So become COOH. Double bonds right in the middle, COOH. That is fumarate. Okay, when you see the fumarate generate, the double bonds right in the middle, that always have something going to happen because it's not stable. So we're going to put something back. So what are we going to put something back? Put the water going to back. Okay, then the water comes back. What did it become? COOH, CHOH, CH2, COOH. This is malate. And I always say, if you like a beer, malic acid, that's where it comes from, the malate. Okay, almost done. Not done yet. Goes here. Here you add it back, but still they're going to lose. So what is going to be the last chemical before we finish? COOH, C, double bonds O, CH2, COOH. Oxaloacetate. Oxaloacetate, okay? Then this comes back. Happens here. Since you have a hydrogen and the proton go where they go? NAD reduced to NADH2. Okay, this is the whole thing is finished. Now we're going to go ahead to calculate the ATP. That's aerobic respiration. And how we do the calculation? NAD, nickel adenine dinucleotides, with 2 proton will become NAD, H, and H plus. I wrote it NAD, H2. How many ATP generate? Like I said, when I was a student, it is 3 ATPs, and you guys are 2.5 because you can think about the oxygen phosphate ratio. So you can do 3 ATPs, or you can do 2.5 ATPs. It's up to you because you calculate phosphate oxygen ratio. Okay, same thing. The bottom one, FAD with 2 proton become FAD H plus and H, which means FADH2. When I was student, it is 2 ATPs, and your textbook is 1.5 ATPs. Now, either one you can use, but you only gonna be stick to one set. I would use this set because it's easy. If you want to use a difficult one, that's okay. If you're already being accustomed to your biochemistry textbook, but don't overlap. You can, the first one use 3 ATP, the second one use 1.5, then vice versa, makes too complicated. Okay, so I stick 3 ATP and 2 ATP, so let's do the calculation. Let's talk about glycolysis. We already talked about, in the 6 carbon area is 2 ATP used. So, in the glycolysis, what happened is used 2 ATPs in the 6 carbon area. In the 3 carbon area, how many ATP generated? 3 substrate level phosphorylation. 1 and 2. Okay, So there is 2 ATPs generated through substrate level phosphorylation. But don't forget, this is a branch. So you have to multiply by 2. Equals 4 ATPs. But do not forget, this is aerobic respiration. This is not glycolysis single cycle. So what this means, this guy here, if it's an aerobic respiration, they connected to electron transport chain and generate ATP. So how many there? Is 3. So that's 3 ATPs, 3 oxidative phosphorylation, of course 2 lines, so 6 ATPs. And you add up together, -2 add 4 add 6 equals 8 ATPs. Okay, that's glycolysis. So I said it one more time. If glycolysis is a single cycle, or it's connected to the fermentation, how many ATP generated? -2 plus 4. Because this guy is not related to the electron transport chain. But if we calculate the total aerobic respiration, this guy gonna be calculated 3 electron transport chain. So that's why there's extra 6 ATPs comes from. That goes through oxidative phosphorylation. So let me help you. 8 ATPs. There is actually 2 ATPs going through substrate level phosphorylation and 6 ATPs generated through oxidative phosphorylation. Now, of course, that is one line of each. Sorry, that's 4 ATPs, not 2. That's 4 ATPs. Okay? Because we used the 2, so -2. Okay. Okay. That's glycolysis, 8 ATPs. Let's look at transition step and the citric acid cycle. We're gonna have to circle that. Because it's aerobic respiration, so it relates to electron transport chain. NAD reduced to NADH2 happen there. Second place happened. What else? This is okay. Okay. Third place happened. And fourth place happened. So we circle. How many ATP generated? So let's talk about citric acid cycle. That is 3 ATPs. This is NAD goes to NADH2. Multiple by 4. 1, 2, 3, 4. So equals 12 ATPs. What else are we missing? FAD reduced to FADH2 have 2 ATP generated. So how many? 2 ATPs generated through FAD reduced to FADH2. Multiple by 2. Sorry, only 1. That equals 2 ATPs. And do not forget the last thing. There is one substrate level phosphorylation happens here. Okay? So there is 1 ATP generated, which is actually GDP become GTP, and this is substrate level phosphorylation. That is 1 ATPs. So how many you add up together? 12 add 2 add 1 is 15 ATPs. And do not forget, we have 2 pyruvate generated, each go to citric acid cycle. So that is 15 ATPs. Multiple by 2 equals 30 ATPs through citric acid cycle. And you have 8 right here, you have 30 right here, you add up together. 8 plus 30 equals 38 ATPs. Okay, I have to emphasize, this is for bacteria. Okay? If fungi, 36. Because 2 ATPs passing the mitochondrial barrier, I said like pay the ticket to go to the gate. That's the whole story. Now let's see how many extra points you're gonna get. I said one name, 0.5. One chemical structure, 1. So how many? 1.5, 3, 4.5, 6, 7.5, 9. Okay? 10.5, 12, 13.5, 15. Is that right? If you want to write that one, it's okay. I'll give you some extra. 15. Okay. Go to here. This one, 16.5, 19, 20.5, 22. Okay. Yeah. 22, 23.5, 25. Then go here, 26.5, 28, 29.5, finally is 31. How many points you added? Add up together.
Amino acids too, right?
Yeah. That alone is how many points? 30 is 32. Amino acids we'll talk about next week. Okay. So overall, and also the 4 gene base, ATGCU, 5. Is that okay? 60 extra points for you. And I tell you one thing, lots of the students catch up. Even they didn't do very well for the exam 1 and the exam 2. And some of you even did very good, only need a little bit of points for the final exam, you get a solid A. So that's beneficial for you. Is that be nice? So you have to study harder. It's all there. Now, I also want to tell you one thing. If somebody said that it's difficult to remember the structure, is that right? At least you know the name. That's not difficult. If you say those structure really hard, can you draw glucose? Can you draw glucose 6-phosphate? And here you at least know citric acid and isocitric acid. There's a couple of easy chemicals you should know. Is that right? Try your best, then you could get at least somewhere 20 points. You made all the lose points before, could catch up here. Okay? That's a key thing. So, make sure you study hard. Okay? So, I will be stopped here and put on YouTube channel.